I'm very thankful. Again, I'm extremely excited to give my presentation on guiding stimulation intensity for RTMS using prospective individualized electric field modeling. So in my presentation, I will argue that at convention, we determine the stimulation parameters and not the properties of the electric field. This can be limiting in certain cases because it is the electric field that mediates the effect of RTMS starting from the subcellular to the functional level. As you already mentioned in the introduction, I will focus on how we determine the stimulation intensity by conventional methods. And then I will show two examples where we use prospective electric field modeling to set the stimulation intensity for RTMS. So for those who are not so familiar with RTMS, so what is it? This is called repetitive transcranial magnetic stimulation. This is an interventional approach which works like the following. You need to buy a stimulator and a coil. The stimulator will inject a very strong current into the coil. This strong but short current, so brief current, will generate a time-varying magnetic field that will enter the scalp and generate intracranial the electric field. I think it's important to emphasize because it's the electric field that mediates the effect of RTMS at different scales, at least according to the present understanding. Therefore, the term magnetic stimulation might be a little bit misleading for those who just started working with this device or technique. RTMS is also an increasingly used stimulation method. We recently performed a systematic review since 1991, which was the first RTMS study published and available on PubMed. And since then, until the end of summer, we were able to find out that there are more than 3,800 RTMS studies, and this is only original human studies, so where RTMS was applied on human participants. So this does not count animal studies or in vitro stimulations. So it's a huge number. So why is this popularity? So RTMS in neuroscience research is used as an interventional approach. You might know that there are two main approaches, observational approaches and interventional approaches. When you use observational approaches, you can characterize the spatial and temporal characteristics of neuronal activity. However, with RTMS or other non-invasive brain stimulation techniques, you can also modulate it. So the basic idea is that the electric field generated by RTMS perturbs brain activity. And by perturbation, I mean simply a reversible but temporal change of brain activity. And I use brain activity here as a very general term, which can refer to many different dependent variables, starting from electroencephalogram measurements or functional magnetic resonance imaging outcome measures. So the idea is that the electric field produced by RTMS perturbs brain activity, and therefore it should also change those brain functions that are dependent on this particular brain activity. So here you have an extra with RTMS and also with TAC as well. I will focus on RTMS in my talk. So here you have an extra opportunity to link brain activity causally to brain functions. Because if you change brain activity and you observe change at the behavioral level, then you can say that the two measurements causally relate to each other. This is a term what we usually use in scientific papers like causal inference, which also requires that you can change the required brain activity in a reliable and well-controlled manner. So according to this logic, the task of the scientist or researcher is to select the stimulation parameters in order to induce the desired effect. However, this is still quite challenging even nowadays. The one reason for this is that we have still difficulties linking the stimulation parameters a priori to the produced electric field. The main reason for this is that we do not have an electric field button on RTMS devices. There is, of course, a good reason for this, because without knowing the anatomy, the brain geometry and the head geometry of the participant, we cannot set the stimulation intensity and the properties of the stimulation in a way that this will produce a particular electric field in the brain. We need to perform simulations, computational approaches in order to get this information. So I state that we set up the stimulation intensity instead of the electric field. So what do we do if we want to set the stimulation intensity? If we do not set the stimulation, the electric field strength, then what do we do? So exactly, we checked this question in a recent review. We did a systematic review from 1991 and 2020. So we selected articles that have been published in this time frame. This is obviously a huge number what we could access through institutional subscriptions. So we took a given percentage of these articles and manually processed them. So what we found was that 90% of the studies used the so-called motor threshold intensity selection approach. So it's either based on the resting motor threshold, the active motor threshold, or the authors might not specify whether this is an active or a resting motor threshold. So how does this procedure work? This is the most frequently used procedure. I assume that most of you are familiar with this, but to make sure that everyone is on the same page. So basically here we've got two main steps. In the first step, you estimate the motor threshold intensity with single pass TMS. And in the second main step, you set or determine the stimulation intensity for RTMS. So what we typically do is first we search for the motor cortical representation of a small hand muscle, for example, the first dorsal or interosseous muscle. And whenever you are at the right spot and use the right stimulation intensity, you can induce small muscle twitches because you stimulate the motor cortical representation of this particular muscle. If you link this to electrodes and then you record it with the computer, you will see a phenomena which is called motor evoked potentials. These are just the evolution of the signal over time. So here you've got the amplitude and here you've got the time. So this corresponds to this muscle twitch. And if the participant's hand is at a relaxed mode, so you don't ask the participants to contract the hand muscle, then you've got, you can measure the resting motor threshold. So the idea is here that if you can evoke these motor evoked potentials in a given number of trials, let's say 5 out of 10 times, so reliably, then you will estimate the minimum intensity which is needed for these muscle twitches. And then we determine the resting motor threshold. If you're ready with this, then what you need to do is to check the stimulator, maximum stimulator output. This is a percentage between 1 and 100. This is an arbitrary number because it depends on the stimulator and the coil characteristics. But all you need to do is to know this number and you are ready with the first step. Then you can transition to the second step. Here, what you need to do is you need to determine the stimulation intensity for RTMS. You might want to use 100% of the resting motor threshold, then you use the same stimulation intensity, or you might use slightly higher or lower intensities. And then the final step, you move the coil to a completely different brain region, which might be characterized by a different cortical folding pattern, different scalp to cortex distance, and maybe this might be on another hemisphere than those where you determine the motor threshold. So the take-home message of this slide is that we determine the stimulation intensity by giving a given percentage related to the resting motor threshold. For example, you write in the article that I used 110% of the resting motor threshold in my experiment. Or if we are lucky, we might also associate this number to the maximum stimulator output maybe you provide a table where you have the individualized intensities according to the maximum stimulator output percentages. But the thing is that here we determine the stimulation intensity not based on the produced electric field strength, but to the motor threshold or to the stimulator's output percentage. Because most studies use the motor threshold intensity, we also checked what are the typical intensities what we use. And we found that this ranges between 80 to 120% of the resting motor threshold. And here comes something interesting. So what we can see here that most of these studies use the same intensity range to induce many different neuronal effects, starting from stochastic resonance to neuronal entrainment, neuronal interference or virtual lesion, long-term potentiation or long-term depression-like effects. And if you check cognitive studies, they use the same intensity range in order to enhance, or the opposite, in order to interfere with brain functions. So by this conceptual, in this framework, we think that the same intensity range can effectively activate many different types of neuronal effects. And you might wonder, metaphorically, that if we use the same intensity, how does the brain know what neuronal effect a given researcher wants to induce? Of course, the common answer is that there are different other parameters of RTMS that can induce the effect, including the stimulation frequency. So you might say that, yes, we use the same intensity, but a 1 Hertz RTMS induces long-term depression-like effect, and 10 Hertz, at least at the corticospinal level, excitability level, or 10 Hertz increases LTP-like effect. Or you might say that the characteristics, the temporal evolution of the pulses, so the interburst intervals, or the or other parameters like the total number of pulse matters. And I agree to all these points, but I think we missed something, because we think that the same intensity range can induce many different neuronal effects. So we treat RTMS induced or TMS induced motor evoked potential, this intensity needed for the muscle twitch, as a universal physiological filter, as Sarah Lisenby nicely coined this term in the recent Baden-Baden meeting on transcranial brain stimulation. I think that measuring the motor threshold is very useful for many cases, but obviously this has some limitations which can be maybe overcome by using other approaches to select the stimulation intensity. And because the motto of this morning session is how to increase the efficacy and reduce the inter-individual variability for transcranial brain stimulation approaches, in the second part of my talk, I try to address these points with some examples and based on our experience with RTMS. So my first suggestion is that consider reducing the electric field strength variability. I claim here that if you use this motor threshold intensity selection approach, it gives the illusion that you standardize the electric field or the stimulation intensity across the participants, because everyone received the same 80% resting motor threshold. However, because the resting motor threshold intensity depends a lot on the participants, this will produce a strong variability in the inter-individual and intercortical electric field measures. This is what I recently did. So I used an open database which has MRI data and resting motor data, resting motor threshold data from the same participants. So this data set allows us to perform electric field simulations for the motor threshold, the fixed intensity, and the Stokes method. I haven't talked about the Stokes method so far. So the fixed intensity means that you use the same stimulation intensity for every participant, let's say 60% of the maximum stimulator output. And the Stokes method, it's another method which was developed by Stokes and colleagues, because this is based on the fact that the scalp cortex distance might be different at the motor cortex as compared to the dorsolateral prefrontal cortex. So a common observation is that we've got a little bit larger scalp cortex distance in the motor cortex than in the frontal regions or the prefrontal regions. And therefore, if you would use the same stimulation intensity, this region would receive a little bit higher stimulation intensity or electric field because due to the reduced scalp cortex distance. So therefore, a possible correction for this is to measure the scalp cortex distance at the motor cortex, as well as your target region, calculate the difference, and apply a scalar in order which will adjust this distance and the stimulation intensity in order to get a roughly similar electric field in this region. So we tested all these methods over the motor cortex and the DLPFC. We selected the motor cortex because in our previous systematic review, we proved that we found evidence that most studies use the 90% resting motor threshold data, the motor threshold stimulation intensity level, and we selected the dorsolateral prefrontal cortex because this is a very common target for many cognitive studies, as well as studies that check the clinical efficacy or protocols in a clinical treatment. So at each point, we created a 3x3 grid location, just simulating the possibility when you might incorrectly adjust the position of the coil of the motor cortex. So at each cortical target, we've got 9 different points where we estimated the electric field. And to determine the motor cortex, we used MRI, like anatomy, determine the central, precentral gyrus. Then we check the scalp location, the corresponding scalp location, and use the 5 centimeter rule, which is a common rule in research as well as clinical applications to determine the dorsolateral prefrontal cortex location. So here we extracted the electric field values corresponding to the gray matter volume compartment. We checked the absolute electric field values, the strength of it. We were using spherical ROIs at each region. We used, in fact, many different spherical ROIs in different sizes just to keep track the effect of the distance from this cortical target. We characterized the electric field strength at the motor cortex and the DLPFC. And from each spherical ROI, we determined the median of from the electric field distribution. We determined these values for the resting motor threshold, fixed intensity, and electric field optimization approach. As you can see here, both conventional approaches produce highly variable electric field strength. Of course, this is a little bit biased by the fact that we've got different locations and different spherical ROI sizes, but the most important thing is that still the inter-individual electric field strength variability is quite large because participants have different resting motor threshold intensities. And if you standardize the stimulation intensity based on the resting motor threshold, then the E field values will be quite different. We've got a very similar pattern at the dorsolateral prefrontal cortex. The variability stayed the same. In fact, we've got slightly higher intensities because due to the scalp to cortex distance. So we also estimated the scalp cortex distance at the DLPFC. This was shorter, and therefore we overstimulated the DLPFC if we would have used the same stimulation intensities. Then we checked the Stokes correction methods. I was quite surprised about this because this actually worked pretty well in reducing these extra intensities in the electric field strength compared to the motor cortex. However, in order to get the inter-individual variability, we needed to perform electric field optimization, computational modeling to get the values right. You still might say that here the E-field values are, the variability is disturbingly large. The reason is that here, for example, for these values, I optimize for the 5 millimeter spherical ROI and not for the other ROIs. And then, of course, if you check the other ROIs, then you still got some variability in the E-field. But if we just particularly check for those ROIs where we optimize the E-field, then you've got virtually identical intensities and very much reduced inter-individual variability. So in order to reduce the inter-individual and intercortical, so between participants and between cortical targets, the variability, then you need to do some sort of computational modeling, because the conventional approaches and their correction methods do not allow you to do so. I also calculated the coefficient of variations and transformed into percentages. And this just reinforces what I described, that in order to get the variability low, you need to do some sort of E-field modeling. So my first suggestion was that you might consider reducing the electric field strength variability by using computational modeling. But the question is whether this can be used in a real experiment. So next, I would like to show two examples where we in fact use this approach, namely that from each participant, we created individualized realistic multi-compartment head models, and we performed prospective E-field simulations in order to set the stimulation intensities for the RTMS session. In the main experiment, each participant took part in a random order in 3 different conditions. And in each intensity condition, the subject received rhythmic and arrhythmic protocols. So the main difference was that in the rhythmic protocol, we've used rhythmically applied the RTMS forces at individual alpha frequency, whereas in the arrhythmic condition, it was pseudo-randomized. So it was not rhythmic, but therefore arrhythmic, but all other parameters were kept constant. In the control experiment, we've got, we stimulated our participants with sham stimulation. So here, sham protocol was applied by tilting the coil by 90 degrees, and we used for each participant 29% of the MSO, because this was the highest stimulation intensity what we had here for one participant in the high intensity condition. Here we only applied the rhythmic RTMS sham protocol. And this is what we found. The analysis was performed by the PhD student of Professor Paulus. She is Elina Zmaikina, and she first estimated the phase locking value for the rhythmic and then for the arrhythmic RTMS protocols in the low, medium, and high conditions, as well as for the sham RTMS. Here you see the normalized PLV values in the alpha frequency range. This X mark represents the location where we place the coil. And here you see the topographical depiction of the PLV changes after the stimulation, during the stimulation. So what you can see here, only by focusing on the Rovereto EEG channels and the individual alpha frequencies, that for both real and sham stimulation conditions, we've got an initial increase, and the phase locking values, which kind of measures the amount of synchronization relative to the ongoing RTMS pulse. So It's a kind of proxy for measuring neuronal entrainment effect. So we found that there was an initial increase in the phase locking values for all conditions, including sham, for which we were extremely surprised because here we tilted the coil, so the E field was much reduced compared even to the low stimulation condition. The only difference was that, or the only common thing was that participants hear this click sound, this rhythmic click sound every time. And this induced this, increased this initial change in the PLV values. But in order to get a significant change to the sham protocols, we needed to apply higher stimulation intensities, starting from approximately 30 and up to 50 millivolt per millimeter electric field strength. Here we also used the gray matter compartment and the absolute electric field strength. Still, we name these components as medium and high stimulation intensity sessions. However, if you check, if we translate it back to the resting motor threshold percentages, because we also estimated the resting motor threshold for each participant, we can see here that still, even in the highest stimulation intensity condition, what we have here in our experiment, the resting motor threshold data was, or the resting motor threshold values were even less than 50%. So it's still considered to be a very low stimulation intensity according to the conventional RTMS intensities. In a second experiment, we chose another cortical region, the motor cortex, and here and stimulated the participant over the hotspot, motor cortical hotspot of the first dorsal interosseous muscle before we stimulated the parietal occipital region, here the motor cortex. And I just wanted to quickly mention this, that she conceptually replicated the findings what we got in the first study. The details of the study is available already as a preprint. So again, for the rhythmic condition, we've got an initial increase as well as like a maintained increase in the phase locking value in the rhythmic main condition. This was not the case for the arrhythmic condition when you do not have this rhythmic application of the pulses, but again, this is pseudo-randomized. And in the sham condition, the pattern of the findings was exactly the same as before. So there was an initial increase, but in order to get a maintained elevation of the amount of synchronization in the mu alpha rhythm, you needed to use main RTMS because sham just probably because participants hear this initial, this click sound, and there is an adaptation to that, this decreased. And so this was not significantly increased anymore. She also compared the rhythmic versus arrhythmic conditions during and after the stimulation, as well as to the sham condition. And we found a significant increase during RTMS and a significant increase only between rhythmic and arrhythmic after the stimulation. So here I showed you two examples where we use prospective electric field estimation in order to individualize the stimulation intensities based on the produced electric field strength. And in both cases, we used very low stimulation intensities and showed that by standardizing the electric field strength across different cortical regions and participants, we could show electrophysiological effects for RTMS. So my last suggestion where I would like to address how to increase the efficacy and reduce the intersubject variability of TBS protocols is rather a practical one. Here you need to apply prospective E-field modeling, but in my third suggestion, this is a suggestion what everyone can do right ahead. And I think it's very important to have the field report enough information which is required to reproduce the stimulation intensities in RTMS research. I think this is one crucial issue, and how we report the RTMS parameters in studies. There are many different parameters. I understand that it's extremely hard to find out which parameters are important to report. And therefore, in our preprint, which is available on bioRxiv, we prepared a kind of not an official guideline, but framework according to which you can maybe check your study in the future to make sure that you report enough information in order to make your article, design your or report your article in a way that the stimulation intensity is reproducible. Because according to our systematic review, based on the 760 articles, we found that only 8% of the studies produce enough information needed to reproduce the stimulation intensity. And the main reason for this was the fact that most people report the percentage of the motor threshold intensity. For example, I say we used 90% of the resting motor threshold, but we do not report the corresponding maximum stimulator intensity in percentages. This is crucial because we need basically two information for retrospective electric field approximation, the maximum stimulator output percentage and device information. So it's also crucial to import stimulator and the coil properties. At least you should report the company and the model number. So if we say we used a maximum figure of 8 coil, we don't know which coil exactly we used, because there are different figure of 8 coils and different stimulators. So it's very useful if you report both the motor threshold, if you use the motor threshold procedure, and the corresponding device parameters. But of course, in order to make it completely reproducible, we need to explain how we ended up determining the motor threshold, because you can estimate the active or resting motor threshold, or you can use another procedure. You can also use different methods. Like the most common is the method of limit, but it has many different versions. Or you can use more complex estimation approaches. You can also assess the muscle twitch visually or using electrodes, as well as you can use different parameters. You can check different muscles, the success rate for determining the motor threshold by using the method of limit, for example, differs. So therefore, it's very useful if you report enough information that can, in which we can reproduce the stimulation intensity as well as the corresponding electric field strength, at least on a template head model. I think this would be also very useful because eventually we use many different stimulation protocols, and it might be the case that this variability, at least partially, due to the fact that we use different electric field strength in different studies. So I talked about the notion that the motor threshold intensity selection approach is the most common approach. This has been used for decades. The main reason is it's quite uncomplicated, and yet it provides individualized stimulation intensities, but externally inducing muscle twitch or phosphines requires strong electric field that might be disrupting in nature. For example, speech rest. It's when you are not able to speak because with TMS, with RTMS, you stimulate the Broca area. However, the purpose in neuroscience as well as in treatment is to induce targeted enhancement, rarely interference. And this is more challenging because we need to get a better knowledge about the involved cellular mechanisms of RTMS. Whereas Lauren Frank says it's very easy to break things in biology, but it's really hard to make them work better. So at the very end, we would like to understand the cellular response to RTMS, and measuring motor evoked potentials is very useful, but this will not lead us to this understanding necessarily, because we need to apply computational and basic science approaches to better understand the neuronal and cellular mechanisms of RTMS. And there, for example, in a brain slice experiment, 90% of the motor threshold is very hard to translate to a useful stimulation intensity. Thank you very much for your attention. And I'm also glad to be part of a bigger team. And currently I'm working in the laboratory of Andreas Vlahos, but I also have a lot of exchange with other collaboration partners like the OPITS lab and postdocs and PhD students in the OPITS lab who are extremely, to whom I'm also extremely thankful, as well as Gregor said, I worked a lot in the laboratory of Professor Walter Paulus. Some of the experiments I showed you was done in collaboration with Elina Zmaikina, who is the PhD student of Professor Paulus, and then Matthias Mittner, who is another collaboration partner in Norway, in Trento. Thank you very much for your attention, and I'm looking forward to your questions.